Hello friends, welcome to my channel. Today we will talk something about determination of amino acid sequence in protein. As you know, when you talk protein, you say that protein is a biomolecule, it is a biopolymer, and it is polymer of amino acids. Okay. So what happened when the amino acids, one amino acid is joined by another amino acid, they are joined by peptide bond, and that is CONH. You can see here, this bond is you call as a peptide bond. This is you say that this is peptide bond, okay? And if you are talking a linear polymer of amino acid, then obviously you say that it is a polypeptide, and you also call it as a primary structure of protein. And you also know that when you talk polypeptide, you are talking that the molecular weight should not be greater than 10,000 Dalton, and above 10,000 Dalton, polypeptide is protein. Okay, obviously molecular weight concentration, and below 10,000 it is polypeptide. So what you can see that in general, primary structure of protein represent linear polymer of amino acid. Okay, that is you call peptides, or you also say polypeptide. Now here you see a tripeptide has been represented. Okay, this is component, one component of amino acid, another component of amino acid, and this is the last one component of the amino acid. So it is a tripeptide. So what you can see, as I have mentioned here, the left side is always will be having N terminal, and the right side will be having your C terminal. Okay, it is designated to indicate the position of NH2 group and carboxyl group. Okay, now question is how to determine the amino acid sequence. That is, which acid is on the left side first, followed by others, and so on. Or similarly, if you take determine the sequence from right side, so what is the first amino acid on the right side and to which it is connected, and so on. So here it is, but it is a very difficult task. We will give example after that. But here is the statement that determination of amino acid sequence constituting a polypeptide. Obviously, when you say polypeptide, the number of amino acid is very more than 4, 5, 6, 7, 8. Is a very difficult task. Okay, and apart from that, as I told you, that the sequence of amino acid can be determined from either side. That is from the N terminal side and from the C terminal side. Okay. If we consider formation of a dipeptide by two simplest amino acid, that is glycine and alanine, so there is possibility that two types of dipeptide will be formed. Okay, one will be having represented by glycine dash alanine ALA, GLY dash LA, and second will be LA GLY. Now you can see here, that is, here is the glycine and this is alanine. So what is happening on combination, they are forming two types of peptide, dipeptide. Now you can see here the N terminal is different. In this case of dipeptide, N terminal is different from N terminal of ALA-GLY. Okay, that is alanine glycine dipeptide. So you can see that similarly C terminal is also different. C terminal is also different. Obviously, when you are talking only and you are talking only 2 amino acid. So what will be the case when you have in polypeptide having n number of amino acid? Well, in that case, the possibility of sequence can be factorial n. That is, a huge number of possibilities there as the number of amino acid increases in a polypeptide. Now, various process has been reported by different scientists to establish amino acid sequences. As I told you that this amino acid sequence in polypeptide can be determined either from N-terminal site, that is amino end, or carboxylic end site. Okay. Now, what happens during course of this amino acid sequence determination? Degradation of polypeptide take place. So the various process can be divided as follows. Now you can see the first process, as I told you, that is amino end N-terminal end degradation method. Okay, and these involves, there are so many methods by which a sequence of amino acid in polypeptide from N-terminal end is determined. The important ones are Sanger's method, it's Dancel method. Dancel method is modification of Sanger method. Okay, then you have Edman method and enzymatic method. Edman method is considered as one of the best method and successful method. Also, you have infrared spectroscopy. By application of this infrared spectroscopy, the determination of sequence, amino acid sequence, has been done. Now, when determination of sequence of amino acid is done by carboxylic end degradation method, the following process has been reported: Shalak and Kampf method, and hydrogenolysis. As you know, that is reaction with hydrazine. It is associated with reaction of polypeptide with hydrazine. This is also called Akbari-Etel process. And also you have reductive method. Obviously, what you do reduction, and you can see when you see the polypeptide, the reduction, when you do the reduction, what you will be reducing? You will be reducing the terminal carboxylic group into corresponding alcohol. Okay, and then you have enzymatic method. Here also enzymatic method. In both cases, certain enzymes are very specific to cleave the peptide bond between two particular amino acid. On the basis of that, you determine the amino acid sequence in the polypeptide also. So these are the various methods which you applied from either from N-terminal side or the carboxylic side. Okay, thank you.